I want you to picture something a little unsettling happening inside your head right now.
Oh, boy.
Yeah. As you are sitting there listening to my voice, millions of tiny spider-like cells are actively crawling through the tissue of your brain.
Right. They are incredibly busy.
Exactly. They are reaching out with these long, sensitive tentacles, palpating your neurons, sensing the environment, and they aren't just sweeping up cellular debris, they are physically devouring pieces of your neural circuitry.
I mean, it sounds like something out of a science fiction horror movie when you frame it that way.
It really does.
But this microscopic process is, well, it's entirely natural. Those spider-like cells are called microglia.
Microglia.
Yeah, they are the resident janitors and immune defenders of your brain. And we're learning that understanding exactly what they are eating could, completely rewrite how we view the aging process.
Okay, let's unpack this. Our mission for today's deep dive revolves around a truly groundbreaking 2026 paper from the journal Neuron.
Right, by Zhang and a whole team of researchers.
Yes, Zhang et al. They have mapped the first ever high-quality single-cell proteome atlas of aging mouse microglia.
Which is a huge deal.
It really is. Essentially, they've figured out how to look inside these microscopic janitors with a level of detail we've never had before.
We're talking unprecedented resolution here.
Right. So we're going to explore what these cells are doing, what they are eating, and the underlying mechanisms of how they malfunction as we get older.
Yeah, the malfunction part is key.
But before we get into the new technology, let's establish the baseline. We know they are immune cells. What is their day job in a healthy brain?
So they are essentially the brain's isolated defense force and sanitation department.
Isolated because of the blood-brain barrier, right?
Exactly. The central nervous system is walled off from the rest of your body's immune system. So microglia have to handle everything on the inside.
Like local security?
Yeah. They defend against pathogens. They swallow up dead or dying cells. And crucially, they perform synaptic pruning.
Synaptic pruning.
Right. Synapses are the physical connections between your neurons.
Yeah.
For your brain to run efficiently, it can't just keep building connections forever.
It would just become a tangled, noisy mess.
Exactly.
Mm-hmm.
So microglia act like gardeners, carefully nipping away the weak, unused, or inefficient connections so the strong ones can thrive.
So they're absolutely vital for a functional, optimized brain. It's like a constant remodeling project.
It is. But the system isn't flawless. Over decades, as the brain ages, these microglia undergo a dramatic transformation.
All right, they change.
They do. They can get stuck in a chronic pro-inflammatory state. Instead of being careful gardeners, they turn into aggressive bulldozers.
Oh, wow.
Yeah, they start overeating healthy synapses, which drives a severe cognitive decline and neurodegenerative diseases we associate with aging.
And that biological problem is what Zhang and the team wanted to solve. Why do they suddenly go rogue?
Right.
But reading through the background of this paper, I realized we've basically been investigating this crime scene using the wrong tools.
Yeah, we have.
Historically, scientists have relied on something called single-cell RNA sequencing to study these aging cells.
And look, single-cell RNA sequencing was a massive leap forward for biology. It allows us to read the mRNA inside an individual cell.
But there's a catch.
There's a fundamental limitation. RNA is just the messenger. It is the genetic instruction manual, the blueprint, that tells the cell to build a protein.
Not the protein itself.
Exactly. It is not the actual protein itself.
So relying on RNA sequencing is like looking at a restaurant's recipe book and just assuming you know what's being served in the dining room.
That is a perfect analogy.
Whereas proteomics, looking at the proteins, is like walking out to the tables and looking at the actual plates of food leaving the kitchen.
Right, because the researchers knew that a cell's blueprint frequently diverges from reality.
How so?
Well, a cell might have thousands of copies of a recipe for a certain protein, but never actually bake it because of some environmental stressor.
Oh, I see.
Or, conversely, it might bake a ton of proteins from just a few recipes. Proteins are the physical engines, the structural bricks, and the chemical weapons of a cell.
They're the ones doing the work.
Exactly.
Yeah.
If you want to know what a cell is actually doing in real time, you have to measure its proteins.
I'm trying to wrap my head around the scale of this, though. I know you can take a tiny scrap of DNA or RNA and run it through a PCR machine to photocopy it millions of times until you have enough to study.
Right, amplification.
Yeah, but you can't do that with proteins, can you? They are physical, three-dimensional structures.
You hit on the exact reason single-cell proteomics has been, well, seemingly impossible until very recently.
Huh?
You cannot amplify a protein.
You just have what's there.
You only have what the cell gives you. And the amount of protein inside a single microscopic microglial cell is infinitesimally small.
So how did they do it?
To measure them, the team used a cutting-edge Orbitrap astral mass spectrometer.
Orbitrap astral mass spectrometer. Say that 3 times fast.
Right. It essentially vaporizes the proteins and sorts them by their molecular weight and charge to identify them.
But getting the cell into the machine without losing the proteins was half the battle, wasn't it?
Oh, absolutely.
They used a technique called FACS, fluorescence-activated cell sorting, to pull the exact microglia they wanted from the brains of mice.
Yeah, they looked at young adults at 2 months old, middle-aged mice at 14 months, and elderly mice at 24 months.
Then they had to deposit these cells into testing plates. And the physics of how they did this is fascinating to me.
The preparation is where many proteomics experiments fail due to something called protein adsorption loss, or PL.
PL, what is that?
Well, when you place a microscopic amount of protein suspended in a liquid into a standard test tube, the proteins naturally want to stick to the plastic walls of the container.
It's like trying to mix a thimble's worth of cake batter in a massive industrial mixing bowl. You're going to lose 90% of it to the sides of the bowl before you ever get it into the oven.
That is the perfect way to visualize it. So to solve this, the researchers deposited the individual cells into specialized, low-binding 384 well plates.
These wells are microscopic, right?
Tiny. By drastically reducing the physical volume of the container and using a surface that repels proteins, they forced the liquid to stay beaded up.
So the proteins had nowhere to stick.
Exactly. Ensuring that when they finally fed the sample into the mass spectrometer, they captured almost everything.
The yield they got is just staggering. They successfully profiled over 3,000 individual microglia across those 3 age groups.
Yeah, it's a massive data set.
And they identified an average of over 1,100 specific protein groups per single cell.
Which proved their method worked. They matched the depth of traditional RNA sequencing, but provided a much more accurate functional picture of the cell's reality.
Let's pause for a second and anchor what we've covered.
Good idea.
Just a quick recap. Bullet 1. We have our brain janitors, the microglia, whose job is to prune our neural wiring. Bullet 2. For years, scientists could only read their genetic blueprints using RNA. But now, we are looking at actual proteins.
The building blocks.
Yeah. Bullet 3. Thanks to massive advancements in mass spectrometry and microscopic fluid management, this research team physically cataloged over 1,100 protein building blocks inside thousands of individual cells.
It's incredible.
It is. It makes you wonder, if you were to look at the gap between what your own cells are genetically programmed to do and what they're actually doing under the stress of daily life, what would you find?
And, that gap between the genetic blueprint and physical reality is exactly what led to the most explosive discovery in the paper.
Let's get into that.
Because they finally had this high-resolution proteomic magnifying glass, they could look at the total inventory of proteins inside the cell.
Right.
And they realized a significant chunk of those proteins didn't belong to the microglia at all.
Wait, really?
Yeah. They coined a new term for this collection, the phagoproteome.
Phago meaning to eat or devour. So the phagoproteome is the physical collection of foreign proteins that the janitor cell has swallowed from its environment.
Exactly.
But wait, if I am looking at a spreadsheet of 1,000 proteins found inside a cell, how do I actually know which ones the cell manufactured itself and which ones it ate?
This is where the team used a brilliant 3-step forensic algorithm. Think of it like an audit.
Okay, an audit.
Step 1. The mass spectrometer flag a highly abundant protein inside the microglial cell.
All right, step 1 is finding the protein.
Yeah. Step 2. The researchers check the microglia's own RNA blueprint library. Does the microglia even possess the genetic recipe to make this protein?
And if the recipe's missing, that's the smoking gun.
Right.
If I walk into your house and find a beautiful, elaborate wedding cake, but you don't own flour, sugar, or an oven, I know you didn't bake it. You brought it in from the outside.
Exactly. But they took it one step further to be absolutely certain of where the cake came from.
Step 3.
Step 3. They take that mysterious protein and cross-referenced it against a whole brain genetic atlas. They scanned the RNA blueprints of every other cell type in the brain, neurons, astrocytes, oligodendrocytes, until they found a match.
So they're essentially opening up the janitor's vacuum cleaner bag, finding a specific type of carpet fiber, and then checking the architectural blueprints of the building to see which room has that exact carpet.
Using that exact logic, The filtering algorithm identified 365 specific outside proteins that made up the phagoproteome.
365.
Yeah. They could confidently say, these proteins were manufactured by a neuron, but we found them sitting inside the immune cell.
So what was in the vacuum bag? What were they eating?
They found a massive concentration of structural synaptic proteins, the actual physical wiring that connects neurons together.
Like SYT1 and SN825.
Exactly. They also found heavy amounts of myelin components, which is the protective fatty insulation wrapped around your nerve fibers.
They caught them red-handed eating the brain's infrastructure.
They did. And to prove this wasn't just a statistical glitch in their algorithm, they performed fluorescent imaging on physical slices of brain tissue.
Oh, they actually looked at it.
Yeah. They used a chemical marker that glows when it attaches to a microglial lysosome.
And a lysosome is basically the cell's acidic stomach, right, where it digests waste.
Right. Then they used a different colored glowing marker that attaches specifically to those synaptic proteins. Under the microscope, they could clearly see the glowing synaptic proteins sitting entirely inside the glowing stomachs of the microglia.
That is so visceral. And how did that vacuum bag change as the mice aged from 2 months to 24 months?
The data showed a terrifying shift. As the brains got older, the sheer volume and proportion of engulfed synaptic proteins skyrocketed.
Wow.
The elderly microglia were hoarding synapses in their stomachs. It paints a picture of immune cells engaging in excessive, reckless pruning as the brain ages.
Let's anchor this again because the implications are huge. Bullet 1. The researchers created a forensic test to determine what a cell has eaten. The phagoproteome.
Right.
Bullet 2. By comparing the proteins inside a cell to its own genetic blueprints, they found over 300 foreign proteins, mostly made up of synapses and nerve insulation.
Mostly from neurons?
Yeah. And bullet 3. Older brains have significantly more of their wiring sitting in the stomachs of their immune cells.
It's a huge increase.
So here's a question for you listening. If your brain's janitors are aggressively eating the connections that store your memories and thoughts, it really frames the brain fog and memory loss we associate with aging in a completely new light, doesn't it?
It changes everything.
It's not just neurons dying, it's the immune system actively dismantling the network.
The clinical context is profound, but we have to look deeper because the researchers found that not all microglia are behaving this way simultaneously.
Oh, really?
Yeah, the cells actually organize themselves into distinct states or tribes, and your ratio of these tribes changes as you age.
So we aren't just looking at a homogeneous army. Some are doing their jobs well and others are going rogue.
Exactly.
The researchers grouped these cells based on their endogenous proteins, the ones they actually made themselves. I want to look at a few of these distinct tribes. Let's start with the perimeter guards, cluster 5.
Right. So the team identified a unique cluster of cells that aren't actually true microglia. They are border-associated macrophages, or BAMs.
BAMs.
They hang out around the blood vessels and the meninges, which is the membrane surrounding the brain. They have a completely different protein signature.
Okay.
And what is fascinating is that they barely change with age. Their protein profile at 2 months looked very similar to their profile at 24 months. They just stand their ground at the border.
But the true microglia deep inside the brain tissue are highly volatile. Let's talk about the healthy state, cluster 3, the youthful pruners.
So this healthy state is highly enriched with homeostatic proteins. These are the molecules that keep the cell stable and functioning normally. But a key detail the proteome Alice revealed was their energy consumption.
Oh, yeah.
These youthful cells had massive aerobic respiration scores.
Meaning they are burning through a huge amount of oxygen and calories, which makes sense mechanically.
Does.
If your job is to physically crawl through the dense matrix of the brain, constantly extending and retracting tentacles to sense the environment, and carefully snip away microscopic connections, you are going to need a massive biological engine. It takes a lot of energy to be precise.
Precision is energetically expensive. But as the brain ages, we see a dramatic shift away from that energetic youthful state and a massive accumulation of cells in a totally different state.
Cluster 4.
Right. A cluster we might call the inflamed seniors, which strongly dominated the brains of the 24-month-old mice.
Let's look at the mechanics of this inflamed state because it's a stark contrast. The homeostatic healthy proteins plummet, and what replaces them?
2 things skyrocket. First, lysosomal markers the proteins that physically build the cell's stomach, like LMP1.
LMP1, right.
These older cells have massively enlarged digestive centers. Second, activation markers, like LGA-LS3. These are proteins that trigger a severe, localized inflammatory response.
I need to challenge a concept here. We keep calling them old and inflamed, but is this just a natural consequence of a cell getting tired and sloppy over time? Or is this a pathological, dangerous state?
That's the big question.
Are they inherently broken, or is something else happening?
That distinction is the crux of neurodegenerative research. They aren't just tired. Their programming is being hijacked by a degrading environment.
Hijacked.
Yeah. The researchers cross-referenced the protein signatures of these inflamed senior cells and found a massive overlap with a known disease state called the microglial neurodegenerative phenotype, MGND.
MGND.
This is the exact signature found in Alzheimer's disease, heavily linked to the immune system's desperate attempts to clear out toxic amyloid plaques.
So they are stuck in a chronic emergency mode.
Precisely. Using lineage tracking algorithms, the researchers mapped out a trajectory. Microglia start in that highly energetic, healthy pruning state, cluster 3.
Right.
But over time, as they're subjected to a lifetime of cellular stress, they transition through intermediate phases like cluster 1 until they get trapped in this bloated, highly inflammatory disease state, cluster 4.
Oh.
They lose their precision, they stop carefully tending the garden, and they start scorching the earth.
Let's take a breath and synthesize this. Bullet 1. We aren't dealing with one type of immune cell. We are dealing with different functional states.
Right, the tribes.
Yeah. You have perimeter guards that stay relatively stable. You have healthy, high-energy pruners that do the delicate maintenance.
And then the shift happens.
Bullet 2. As you age, a huge percentage of those healthy cells mutate into an inflamed, bloated state that shares a biological signature with Alzheimer's disease.
The MGND phenotype.
Exactly. So here's a question for now. Imagine your own immune cells losing their intricate programming, getting trapped in an emergency response, and bathing your delicate neural networks in chronic inflammation. What kind of toll would that take on your local brain environment? The collateral damage would be devastating.
It is devastating, but the severity of that damage depends entirely on where you look in the brain.
Location matters.
Big time. The team didn't just dump all the brain tissue into a blender. They meticulously tracked the zip codes of these cells. They compared the hippocampus to the prefrontal cortex.
The hippocampus is the seahorse-shaped structure deep in the brain that handles memory formation. And the prefrontal cortex is the very front of the brain, right behind your forehead, which handles complex decision-making and executive function.
Exactly.
So do the janitors age the same way in both neighborhoods?
They absolutely do not. The regional dynamics were striking. The hippocampus remained surprisingly stable. Yeah, as the mice aged, the overall ratio of youthful pruners to inflamed seniors didn't swing wildly. The memory center maintained a relatively balanced immune environment.
But the prefrontal cortex was a disaster zone.
The data showed a massive accelerated shift in the prefrontal cortex. By 24 months, the vast majority of the microglia in the front of the brain had plummeted into that inflamed, aged state.
That's wild.
They were aging vastly faster than their counterparts in the hippocampus.
And it wasn't just their state that was different, it was their diet. The phagoproteome, the contents of their vacuum bags, was completely different depending on their zip code.
When they analyzed what the cells had eaten, the prefrontal cortex microglia had engulfed significantly more synaptic wiring.
More SYP and SNAP25.
Yes, and not only that, they were heavily laden with engulfed tau proteins. MAPT.
Tau proteins. That's a big buzzword in dementia research.
It is. Tau is a structural protein inside neurons that, when it misfolds, forms the toxic tangles famously associated with Alzheimer's and other dementias. The prefrontal microglia were gorging on it.
I'm trying to work out the mechanism behind this. Why is the front of the brain aging so much faster? Are the janitors up there just genetically predisposed to failure, or is something else driving their rapid decline?
It almost certainly comes down to their workload. The prefrontal cortex is the seat of complex cognition. Every time a mouse or a human learns a complex new rule, adapts to a new social dynamic, or makes a difficult decision, the prefrontal cortex is actively rewiring itself to accommodate that new behavior.
Which means it generates a massive amount of structural waste. If the front of my brain is constantly building and breaking connections to help me navigate my daily life, the janitors up front are working double shifts compared to the memory center.
The metabolic demand is staggering. Over a lifetime, that continuous heavy requirement for synaptic remodeling pushes the prefrontal microglia to their limits.
They just burn out.
Exactly. They are tasked with clearing so much debris and remodeling so many synapses that they simply burn out. They accumulate toxic proteins like tau, their stomachs become constantly engorged, and they transition into that stressed, inflammatory state much earlier than cells with a lighter workload.
Let's do one final synthesis of these findings. Bullet 1. This study reveals that the geography of your brain dictates how fast it ages.
Location, location, location.
Bullet 2. The decision-making center in the prefrontal cortex forces its immune cells to work at such a high capacity that they burn out, bloat with toxic proteins, and shift into an inflammatory state much faster than the memory centers.
Right.
So, knowing that the very act of complex problem-solving creates an immense physical burden on your brain's immune system, does it make you think differently about how cognitive fatigue develops over your lifetime? It really puts the concept of mental fatigue into a stark biological perspective.
It provides a very physical mechanism for why executive function, our ability to focus, plan, and regulate our impulses, is often one of the first cognitive domains to become fragile as we get older. The cells responsible for maintaining that hardware are exhausted.
If we pull back and look at the whole picture, the value of this neuron paper is just astronomical.
Truly.
By leveraging single-cell proteomics, these researchers didn't just guess what our brain's immune cells were capable of based on their genetic blueprints. They physically audited them.
They checked their stomachs to see what they were actually eating.
Exactly. And they proved that as we age, our cellular janitors lose their precision, become overwhelmed by the sheer volume of brain maintenance, and start recklessly dismantling the neural networks that make us who we are, especially in the areas that govern our highest level thinking.
And beyond the microglia themselves, this data set serves as a new foundational atlas for all of neuroscience.
A new baseline.
Yeah. It proves beyond a shadow of a doubt that relying on genetics or mRNA alone is insufficient, especially when looking at the immune system where the environment dictates behavior. If we want to understand the reality of neurodegenerative disease, we have to follow the proteins.
Right now, in your head, there is an invisible, microscopic battle for balance taking place. Your microglia are sweeping up your neural pathways, deciding in real time which of your synapses to protect and which ones to devour.
It's happening right now.
But if science has advanced to the point where we can crack open these cells and read the exact menu of what they are eating, how far are we from being able to rewrite that menu?
That's the million-dollar question.
Could we one day take a pill, something that reaches into the prefrontal cortex and tells our exhausted microglia to stop eating healthy synapses, calm their inflammation, and specifically target only toxic Alzheimer's plaques instead?
It is a wild frontier.
It really is. Keep questioning the microscopic universe inside your own head, because those tiny janitors are actively writing the physical story of who you are. Thank you for joining us on this deep dive.